Okay, we're back to this. So, yeah, so there will be a, yes?
When were the videos posted?
A week and a half ago? So, I haven't done this in a while. Forgot how to lecture. Quizzes, that's where it's at. Okay, so yeah, quiz will go up on Wednesday. It'll come due on Friday. And then next week there will be a quiz on Wednesday again, and it'll come due on Friday. That will be the end of all that. You have those two things, and you have final. And that's all you have left to do. Yeah. Yeah, the final is going to be cumulative. Yes. It will be designed, it will be slanted towards the most recent stuff. And so it will not be testing equally over all of the material. It will be more heavy on the stuff from between the last test and the final. But I am not designing this final to take any longer than a normal test. It will be the same number of questions. It will be the same distribution of questions. I'm not designing it to be any longer, so it should be about the same. Now, every year I say that, and then it ends up like people take the time they have to take tests. So, yeah. Any other questions about that? Then, oh, yeah. When's our final? I believe that.
Wednesday.
I do not remember.
Tuesday, December
It's
11th
something.
at 10.
I will probably look that up on Sunday night again. Like, be like, oh, do I have to do that tomorrow? No. Yeah. It's sometime during Brown week. I believe that. If I put it in the syllabus, there's a 75% chance that's right. Okay, so actually probably a little higher than that. I think I get right more than 75% of the time. It's probably like 90%. Probably being right. If I have screwed that up before. So it wouldn't hurt to like double check yourself. Okay, so let's talk about lysosomes and peroxisomes. Moving on in these. So let's start with lysosomes. And particularly, so lysosome itself means hydrolytic particle. It's Greek for that. And they're small, they're about 0.2 to 0.5 microns in diameter. So these are relatively small organelles. Hold on till we get to protocatenaries. I have all sorts of cool things to tell you now about protocatenaries, but it's nothing to do with this. I don't know why I said that. Okay. So, and what they do is they're going to be very useful in essentially breaking things down. Hence, lysosomes. They are lysing things. They are hydrolyzing things. And they contain a lot of different hydrolase enzymes. So somewhere in humans, between 40 to 50 different kinds of hydrolase enzymes are going to be in the lysosomes. And this makes them capable of breaking down macromolecules. Chair out of my way. I hate chairs in my way. There. I can move. They're kind of like the garbage disposals of the cell. If you were going to break down an organelle, for instance, you turn to the lysosome. If you were going to break down something that you phagocytosed, you're going to use a lysosome. That's essentially what they do, is breaking down things. So, most of these hydrolases are going to have an optimal activity at a fairly low pH, especially considering what physiological pH is, somewhere between 4.5 or 4.8 and 5.5. So inside the lysosome is a fairly acidic environment. And we have proton ATPases that are extremely important on the membrane of the lysosome that are using energy to pump protons into the lysosome to maintain that appropriate pH. Now, one of the things that's advantageous to having this pH is that you can denature oftentimes proteins at this pH and make them easier to break down. Give them more sites at which these hydrolases can access the proteins, essentially chew them up. It's following kind of a similar principle to your stomach. One of the reasons that you have a low pH in your stomach isn't necessarily to directly break things down, but to denature proteins so that other things then, so proteases can then attack proteins and break them down. So, they're going to be really important in the degradation of endocytosed material. So things are taken up by endocytosis. So this can be anything from macromolecules to bacteria or other things like that. Just random stuff. So let's talk just briefly about the process of endocytosis. And it's going to be similar to something that you saw in the videos, particularly the formation of vesicles, because that's essentially what we're doing. Move on. Come on. Oh. So let's talk about the process of endocytosis. And generally this starts with something in the extracellular area binding to a receptor on the surface of the cell. You have to be able to recognize that there is something outside that you want to endocytose. And that generally happens by something binding to a receptor, whether this is a particular cell surface glycoprotein on a bacteria or some particular macromolecule or something like that binding to a receptor on the surface of the cell. And what you get is the formation of a coated pit on the cell. And that pit is going to be coated with a protein that we've talked about already, clathrin. And so this is known as clathrin-mediated endocytosis. Again, clathrin being a coating protein that's helping you form a vesicle and helping to form that and stabilize that as you actually form the physical structure of that vesicle. And then what happens is that you're going to get the vesicle forming, and that's forming as around whatever is on the outside of the cell that you want to endocytose. Once the vesicle forms, the clathrin is going to unbind, and this is then the endocytic vesicle, or sometimes called the endosome, or essentially particle inside, or the phagosome, which is essentially the eating particle. Specifically, these names are referring to the vesicle after it has shed the clathrin. Let's take a quick look at how this works. Wrong way. Okay, so again, you have something out here that's going to bind to a receptor. Some particles, something else like that that's going to bind to a receptor. And that is going to begin, that receptor is then going to begin a process on the inside of the cell in which we have various coating proteins that are going to begin to form this pit, and then you also have clathrin beginning to form the vesicle as well. And I'm going to not give you all the details in here, but essentially you have this continuing to go until you have it buds off of the membrane itself, and then you have a clathrin-coated vesicle here, which then the clathrin falls away, and you just have the vesicle that is left, with whatever was binding to the receptor in here. And so this, at this point, would be the endocytic vesicle. And so what you have is a primary lysosome. Now, a primary lysosome is just in difference to what we call secondary lysosome. It is a lysosome that has not bound with the endocytic vesicle yet. But you have a primary lysosome, and the endocytic vesicle are going to combine. They're essentially going to meet, and the same way that we talked about vesicle fusion with the membrane happening, you're going to have something similar happening here, except instead of having the membrane being like the cell membrane or an organelle membrane, you have the membranes of two vesicles coming together and combining, but forming one larger vesicle that is now the secondary lysosome, because we have now combined, essentially dumped the contents of the lysosome into our endocytic vesicle, and now we're allowing all of that stuff, all the hydrolases, to get at whatever we endocytose and begin to break that down. It's kind of nice because then during the entire process, we're keeping all of those hydrolases that need to be at a very low pH. We're keeping them at a low pH. We're not exposing them to the rest of the cytosol that would then raise the pH somewhat. We are also not exposing the rest of the cytosol to those hydrolases that, I mean, honestly could break down whatever was in your cell probably, not just the thing that you want. So by combining those two vesicles together, we're dumping all those hydrolases directly into the endocytic vesicle that we want to actually digest things in. And at this point, wastes are degraded. We can take the basic parts of this and recycle them. We can discharge them back out into the extracellular space. If they're proteins, just basic amino acids, we can use those to create new proteins now. If it's something else that we don't want, we can dump it out of the cell. And part of that is going to be determined based on what type of thing was endocytosed. One of the things I'm not specifically talking about here at the moment, but I can go back to it just for a second. Here, these receptors specifically are generally going to be part of a lipid raft. That you have receptors here, but then there are those who are going to be associated with other types of membrane proteins that will then help direct the endocytic vesicle to do the specific thing that it needs to do. And so we're not necessarily just talking about just receptors here. There is a whole complement of similar, or not necessarily similar, but a suite of proteins, membrane-bound proteins that work together for a particular function. They're bound together in this lipid raft that will allow this endocytic vesicle, when you fully form it, here, to have all of those same types of proteins, or all those proteins that you want here, to actually direct where this goes. There'll probably be a suite of other receptors and other things like this that will then cause this to either then rebind with the cell membrane, or perhaps bind with an endoplasmic reticulum, or some other thing like that for further processing or ejecting of that particular waste. Okay. Okay. Now this is actually kind of interesting. This is a very common type of system. The clathrin-mediated endocytosis is this really basic form of endocytosis that is used throughout life. Essentially all living things will use this. And because this is the case, a lot of different diseases have, and a lot of different pathogens hijack this, like specifically have evolved to hijack clathrin-mediated endocytosis. If you are a bacteria and you want to get inside a cell, or a virus, and you express the correct proteins that will cause endocytosis, and then you can either prevent the binding to a lysosome, or you can survive the hydrolases that then get released into the lysosome, you just got free entry into a cell. Woo! And so there's a lot of different viruses and bacteria that will actually end up invading cells through the clathrin-mediated endocytosis route. Some of these include bacteria like Listeria, Yersinia. Yersinia is an interesting one. Does anyone know what Yersinia pestis causes? That is the Black Plague. Yeah. So that's how that gets in. Salmonella. Some ones that are like really like, yeah. Salmonella gets into cells this way. Neisseria, which causes gonorrhea, but then also some kinds of meningitis, also gets into cells this way. But also a lot of viruses. Influenza gets into your cells nucleothrin-mediated endocytosis, Ebola, SARS, hepatitis C. There's a lot of things that take advantage of this pathway to gain access to cells because it is ubiquitous, it's all over the place, and although the receptors are going to be different depending on what kind of organism that you are, the basic functionality of this is different, or the same. Why in the world did I say different when I meant the same? Okay. So, lysosomes mediate a lot of different cellular digestive functions. Since we're talking about cellular digestive functions, let's talk about a couple of different digestive functions that happen on the cellular level. One of them being autophagy. And this is the destruction of organelles or some sort of intracellular molecules. These are damaged, no longer needed, something like that. They're old. There's a whole list of reasons. Most of your organelles are going to turn over at a certain rate faster than your individual cells are. There's a whole list of reasons why this happens. Mitochondria just become damaged, oftentimes because they're working like all the intermediates, the electron transport chain, are really, really active, like really energetic, and they just damage things on accident. So we have organelle turnover happening at a fairly high rate, and this is all taken care of by autophagy. So a nucleus is going to last about 5.1 days, and mitochondria somewhere about 6.8 days. Most of your cytosolic proteins are going to last about 5 days. Your plasma membrane actually goes really fast. It is turned over about once every 2 days in a cell. And so a lot of these individual things in a cell are being turned over at a fairly high rate. And only the actual components of them can then be recycled to make new versions of these. Now, there's a couple different types of autophagy that occur. First off, we have chaperone-mediated autophagy. Now, this is very similar to what we talked about earlier when we were talking about the destruction of proteins in the rough endoplasm in particular. But that's one very specific route. That's not the only way that we break down proteins. Another way that can happen is that it can happen through the lysosome. So individual proteins can be tagged by chaperones and then transferred to the lysosome for destruction and actually broken down into the lysosome. So this is another path of protein degradation that's not the ubiquitin-mediated protein destruction pathway. We also have microautophagy. Now this is particularly where the lysosome is going to envelop and consume part of an organelle or part of small cytoplasmic regions or something like that. We are not destroying an entire organelle. We are destroying part of an organelle that might be damaged or something else like that. And so this is microautophagy. In contrast to that, yes?
What flavor is that?
I'm not totally sure. It's an herbal tea. It has like a little bit of orange in it. It has some like fruity stuff in that. Like some lemongrass in there. I'm not exactly sure what all it has. There's mint. Mint is the major base to it, and then there's other like random things.
Citrus breeze?
No, it's something else. The citrus is pretty mild in there. Anyhow, okay. So in contrast to this, we also have macroautophagy, in which we have a whole organelle or some large cytoplasmic region that is going to be destroyed. And what ends up happening is we wrap around a double membrane, in which we're not exactly sure where that comes from, but it could come from the rough endoplasmic reticulum. I get really nervous about these, like we don't know where this is, and so most years, like I'll do a quick Google search and like, do we still not know? Because I'll feel really dumb if I say we don't know. Some Google says like, no, we know. Okay. Anyhow, beside the point. And what this is forms this structure called the autophagosome. Phagosome, like we talked before, particle that eats something, and auto being like you're eating yourself, or an autophagic vesicle. And this is then going to fuse with a primary lysosome, or sometimes multiple primary lysosomes, and begin to break down that whole organelle. So that's autophagy and several different types of autophagy, but you oftentimes don't only want to break down yourself. There's other things. And so oftentimes we can also digest extracellular materials by secreting enzymes from the cell into the extracellular space. And so we can get breakdown of excessive material in the extracellular space. Stuff on the surface of the plasma membrane. One of the ways that this can happen is by binding of a lysosome to a membrane and dumping the contents of the lysosome into the extracellular space. However, that is oftentimes, that's not the most common route in which this happens, not with just complete lysosomes, because that is a blunt tool to be like just dumping into the extracellular space often. But you also have vesicles, as we talked, from the Golgi that have packaged up some certain protein, some certain enzyme. And so we have either a single type of enzyme or a small class of enzymes that are packaged up in vesicles that come from the Golgi, and then we can bind those with the cell's membrane, dump those into the extracellular space, and actually cause the breakdown of whatever that is that we want to break down. Yes?
Is there an example of like tissue where this would take place? Like some kind of organism?
So this actually happens a lot. One that comes to mind is osteoblasts in bone. And so you have two different cell types that are in bone that are constantly remodeling bone. You have osteoclasts and osteoblasts. Osteoblasts are actively building bone while the osteoclasts are breaking down bone. So osteoclasts, actually both of these will dump things, but the one that's more breaking down is you have osteoclasts that are dumping certain kinds of enzymes that break down both the protein matrix, but then also the hydroxyapatite that makes up the bone. And they work to end up breaking down bone while you have osteoblasts coming behind it and building it back up. And this way you have remodeling of bone that's happening in a fairly regular cycle. Does that make sense? And so they're mostly, like you have these cells that are mostly working to maintain a certain type of extracellular material. Bone. Now that's actually fairly common throughout a lot of connective tissue, that you have cells that their primary function is to maintain a certain type of extracellular material. So another one is like tendons and ligaments is mostly extracellular. Most of the stuff that makes them worker extracellular. Anytime that you have a connective tissue that is exceptionally important, well, anytime that you have a connective tissue, you have most of the structure of that is being external to the cells themselves. And most of those connective tissues then have cells that remodel it on a fairly regular basis. Sometimes those functions are in one single cell type, and sometimes they're in two different cell types. And will end up either dumping out things to break down the connective tissue or to rebuild the connective tissue. But both it's kind of going through a similar situation even if you're building it back up. Another one that is well, I won't do that one. Yeah, so bone is the one that jumped immediately to mind. Any other questions on lysosomes? Yes?
By enzyme secretion, so the lysosomes secrete the enzymes, just like up, or they will.
So the lysosomes can help secrete the enzymes by actually binding to the cell membrane.
Okay.
And essentially they dump their contents into the extracellular space. Yeah. Okay. So let's talk a bit about peroxisomes. Anyone remember any time that we talked about peroxisomes before this point in this class? Carbon fixation? What specifically about carbon fixation?
Plants,
Plants.
we take it if there's to fix photorespiration.
Yeah.
Outside, and we take it to the left.
Yes, exactly. So one of the things that the peroxisome does is help ameliorate the consequences of photorespiration in plants by helping to recycle the phosphoglycolate that we create as we go through and we accidentally make that during the Calvin cycle when Rubisco binds oxygen instead of carbon dioxide. So that's one of the places it came up already. So they're very similar to lysosomes, but they tend to be about a little bit smaller, about 2 micrometers in diameter. So not like orders of magnitude smaller, but a bit smaller than lysosomes. And the thing that distinguishes them from lysosomes is that they have an enzyme crystal at their core. And so a particular, like, you have a bilipid membrane in a peroxisome, and then you have like this crystalloid core that is most commonly present in the center of the peroxisome. So the place that you have most of these invertebrates is you have a lot of them in your liver and a lot of them in your kidneys. The most well-studied functions of them are in the liver. In plants, they're most commonly in the leaves and in the seeds. In the leaves, they take on a big function of trying to recycle phosphoglycolate. In the seeds, they're helping to, with a lot of synthesis of various things. They're also really common in a lot of unicellular organisms. This is a common, there's a lot of interesting things in a lot of unicellular organisms, which we're going to talk about in a few minutes. Yes?
But there's only like the 3 categories they're found in, not in like fungi or stuff like that.
Say it one more time.
Are those the only like 3 big categories they're found in, not in other things like fungi or stuff like that?
So they're also found in fungi and other things. These are the major places that we know of them and have a really extensive knowledge of their function. One of the weird things about peroxisomes is they do a lot of different things. Like there's not like a good, we have some structural things that we say, okay, this is a peroxisome because of these structural things, and also based on the contents of like what particular kinds of enzymes are present inside the peroxisome. But they end up doing this huge, wide diversity of functions. It's not like they all have this one function. Like obviously they're not recycling phosphoglycolate from photorespiration in humans. And so these are the places in which we have a really good idea what they do. They are present in essentially all living things. I don't know if all eukaryotes have peroxisomes. So let me back up on that. All eukaryotes, as far as I know, have peroxisomes. I do not know that you have them. I don't think you have them in prokaryotes, but I could be wrong about that. Okay. So peroxisomes appear to originate from the endoplasmic reticulum. But they also receive vesicles from the mitochondria. They're receiving vesicles from the mitochondria, and we're not exactly sure what the function of those vesicles that they're receiving from the mitochondria are. That's one I didn't. Hold on. There was a 2017 paper published, The Peroxisome Mitochondria Connection, How and Why. Okay, so this might have been a bad idea for me not to Google that one. Okay, I might be wrong about the function is unknown. I'm not going to read this right now because that would be boring for you to all watch me read. Maybe it would be very exciting, I don't know. I could read really oddly, I have no idea. What's that? Okay, so, no, we're not going to do that. So, peroxisomes, they do a number of unrelated things, like I was just talking about. Number one that we've talked about already is helping ameliorate the consequences of photorespiration in plants. This is primarily what they're doing in the leaves of plants. Another big one that they do, particularly in plants and fungi, is beta oxidation. They carry out many of the functions of beta oxidation, where specifically and also breakdown of very long chain fatty acids in animals. So we're talking about breaking down very long fatty acids that are C22, like 22 carbon fatty acid chains and larger. And it actually appears that this is one of the really major things that they do in animals is break down very long fatty acid chains. In fact, there was one disease that appears to be primarily related to this in humans called Zellweger syndrome. This is where a person has few or no peroxisomes, and this leads to a buildup of these very long chain fatty acids and causes a whole host of developmental problems, including enlarged livers, visual impairment, inability to move, and people that have this generally do not make it past a year of life. So peroxisomes are kind of important. Mm-hmm.
The treatment for that, would they try and like implant peroxisome?
I'm not exactly sure what the treatment is for Zellweger syndrome. It seemed, just going off first principles, it seems like it would be an easier feat to try to artificially remove the very long chain fatty acids than to implant peroxisomes into a cell. But I am completely ignorant about that. I'm not exactly sure what the treatment is for that. Peroxisomes are also involved in gluconeogenesis, so essentially making glucose from non-carbohydrate starting molecules like fats, amino acids, lactate. There's a couple more things in this list, but I'm going to leave it there for now because I won't be able to get through all of them. It's about that time anyhow. Enjoy the rest of your day. Have fun being back after being gone for a while, or at least not having to come to class for a while. What's that?